This lesson covers the topic of pigments and the process of photosynthesis. This lesson was designed by Sam Drupp as part of the National Science Foundation-supported Boat of Knowledge in the Science Classroom. Why are flamingos pink? Take 30 seconds to formulate a hypothesis. Share your hypothesis with your neighbors. Flamingos are actually gray. The pink color comes from the carotenoids, a type of pigment found in plants, that the flamingos consume. And we'll be learning about pigments and photosynthesis. First, we'll set up a short experiment looking at the different pigments that are found in a spinach leaf, using leaf chromatography. First, draw a line on your filter paper with the pencil. This line will mark where the pigments will start so you can get a better idea of how fast they're traveling up the paper. Next, transfer the pigments from the spinach leaf to the chromatography paper using a penny. Rub the penny back and forth to transfer the pigments from the spinach leaf onto the chromatography paper. You might have to do this multiple times to get enough pigment onto the paper, or use multiple leaves. Once the pigments are transferred to the paper, you will then suspend the chromatography paper in the solvent using a small amount of tape and the pencil. The solvent is acetone, and you want to make sure when you put the chromatography paper into the beaker, the pigment line or your pencil line does not go below the level of the solvent. Suspend the paper in the middle of the beaker, making sure that it doesn't touch either side. Pigments. Plants have two types of chlorophyll used in photosynthesis, chlorophyll A and chlorophyll B. Chlorophyll A is the primary pigment used in photosynthesis and is common to all photosynthetic organisms, from trees to microscopic algae. Chlorophyll A absorbs red and blue wavelengths. Chlorophyll B is considered an accessory pigment, but can be found in many land plants. Chlorophyll B is very important for plants grown in shady environments, as it allows for the plant to use a wide range of wavelengths. As with chlorophyll A, it absorbs blue light, but also absorbs yellow and orange light. Both chlorophyll A and chlorophyll B reflect green light, which makes them appear green. Plants grown in full sun tend to have leaves that are a brighter green, while plants grown in shade tend to have a more olive color. This difference in appearance is due to chlorophyll B content. Leaves grown in the shade have more chlorophyll B and thus appear darker. Carotenoids are a type of accessory pigment. Carotenoids are xanthophyll and carotenes. They are common in most land plants, and they absorb green and violet light. They reflect orange light, which makes them appear orange. Scientists believe that carotenoids protect chlorophyll A from photooxidation, a type of damage caused by the sun. Chlorophyll A is very expensive, and plants invest energy in creating carotenoids to protect the chlorophyll A. This figure shows the amount of light that can be absorbed by three main types of pigments found in plants on the y-axis. The wavelengths of light on the x-axis. As you can see, each type of pigment has wavelengths of light where absorbance is optimal. There is very little overlap, and by having more than one pigment, a plant can increase the amount of light it can capture and use in photosynthesis. So why are flamingos pink? Flamingos are pink because the food they eat has high concentration of carotenoids. This red color is seen in many different types of animals, but carotenoids can only be synthesized in plants. This is true for the puffin on the bottom left, the salmon in the upper right, and the sea bug, or lobster, on the bottom right. Let's look at our chromatography paper. If the pigments have not traveled at least halfway up the chromatography paper, allow the experiment to continue. We are able to separate the pigments on the chromatography paper due to differences in size of the pigments themselves. As the solvent moves up the paper, it grabs the molecules found in the spinach leaves. The smaller pigments move faster than the larger pigments, causing the pigments to separate as the solvent moves up the paper. When you look at the chromatography paper, you can see 3 different lines of color. The first, brighter green, is chlorophyll A. The second, the darker green line, is chlorophyll B. And lastly, we can see a faint orange line. These are the carotenoids. You can do this experiment again with different types of leaves and compare your results to the spinach leaves. There are 2 main types of organisms on Earth, autotrophs and heterotrophs. Autotrophs convert light energy into chemical energy, where heterotrophs must ingest food to obtain energy. All energy available to living organisms on Earth comes from the sun. Autotrophs use the process called photosynthesis to convert light energy into chemical energy or sugar. This is the chemical reaction of photosynthesis. Carbon dioxide, CO2, and water, H2O, are converted by the plant in its chloroplast to oxygen, O2, and glucose, C6H12O6, using energy from the sun and a suite of intracellular enzymes. Photosynthesis occurs in the chloroplast. The chloroplast is an organelle within the plant and some protocells where photosynthesis occurs. Within the chloroplast, there are structures called thylakoids. Stacks of thylakoids are called granum. Photosynthesis occurs in the membrane of the thylakoids within the chloroplast. In the light-dependent reaction of photosynthesis, water is split into oxygen, O2 gas, and hydrogen, H+, and an electron. The electron is then excited by the energy captured by chlorophyll A in photosystem 2. The energized electron then travels through the electron transport chain to photosystem 1, transferring a second hydrogen ion through the thylakoid membrane. The low-energy electron is then excited again in photosystem 2 and used to create NADPH, a type of cellular energy. This process also creates an imbalance of hydrogen ions on the inside of the thylakoid membrane, in the thylakoid lumen. To return to homeostasis, hydrogen ions leave the thylakoid through the enzyme called ATP synthase. As the hydrogen ions move through ATPase synthase, the structure rotates, much like that of a water wheel, generating ATP, the primary type of cellular energy. The light-dependent reaction occurs in concert with the light-independent reaction. The ADPH and the ATP generated in the light-dependent reaction are then shuttled to the light-independent reaction where carbon dioxide is converted into sugars, a type of long-term energy storage. If you were a scientist trying to measure photosynthesis, you could easily measure the amount of sugars produced in the light-independent reactions or the amount of oxygen produced in the light-dependent reactions to determine photosynthetic rate. We will determine photosynthetic rate using the floating leaf discus experiment. First, use a hole punch to punch out 10 discs from spinach leaves. You might have to use multiple leaves. Remove the plunger from the barrel of the syringe and place the discs into the syringe barrel. Replace the plunger. Pull in around 20 milliliters of buffer solution. Turn the syringe upside down, make sure all of the leaves are floating, and then expel the air out of the top of the syringe by pushing down on the plunger. Once all the air is out of the syringe, put your thumb over the opening of the syringe, pull back on the plunger, and shake vigorously. What you're trying to do in this step is remove all of the air from inside this structure of the leaf. You might have to do this multiple times, but eventually the leaf should start to sink. Once all of your leaf discs are sinking, empty them into a beaker full of the solution and place them under direct light. Once you place the leaves discs under direct light, the leaf discs will start to photosynthesize. As photosynthesis occurs, the amount of oxygen in the discs increase and the disc begin to float. You will watch your leaf discs and count the number of discs floating every minute for the next 15 minutes. At the conclusion of the experiment, you can then determine photosynthetic rate by plotting the number of discs floating over time.